Hello students, today we will study about the microsporogenesis and megasporogenesis with the help of permanent slides. So the microsporogenesis is the process of formation of microspores or pollen grain from a pollen mother cell or microspore mother cell through meiosis. So this process is known as microsporogenesis. Microsporogenesis is studied under two heads. Number one is structure of anther. The fertile portion of the stamen is called anther. Each anther is usually made up of two lobes connected by a connective. In turn, each anther lobe contains two pollen chambers placed longitudinally. Each pollen chamber represents a microsporangium and is filled with a large number of pollen grains or microspores. The pollen sacs are surrounded by four layers, as you can see in the photograph. Number one is epidermis. This is the outermost single layer and protective layer. Next one is endothecium. Inner to the epidermis, there is a single layer of readily elongated cells. The cells of endothecium develop fibrous thickening, which help in the dehiscence of the anther. Next layer is middle layer. 3 to 4 layer of thin wall cell situated just below the endothecium is known as middle layer. Cells of this layer are ephemeral and degenerate to provide nourishment to growing microspore mother cell. Next one is tapetum. This is the innermost layer of the wall. The cells are multinucleate and polyploid. Tapetal cells are nutritive. In these cells, Ubis bodies are present which help in the ornamentation of the microspore wall. The tapetum is of two types, amoeboid type tapetum or secretory or granular tapetum. Now, next is megasporogenesis. The process of formation of megaspore from the megaspore mother cell is known as megasporogenesis. So now let us discuss about the structure of ovule, that is megasporangium. Ovule is considered to be an integumented megasporangium. The ovule consists of stalk and the body. The stalk is called funicle. One end of the funicle is attached to placenta and the other end to the body of the ovule. The point of attachment of funicle with the body is called hilum. Sometimes funicle get fused with the body of the ovule one side and form a ridge known as raphe. The body of the ovule shows two ends, the basal end, often called the salazal end, and the upper end is called micropylar end. The main body of the ovule is covered with one or two envelopes called integuments. These leaves an opening at the top of the ovule called micropyle. The integument encloses a large parenchymatous tissue known as nucellus. The residual part of the nucellus in the mature seed is called perisperm. In the center of the nucellus is situated a female gametophyte known as embryo sac.